So thank you for giving me the opportunity to talk. I would like to discuss the impact of odr-3 and ife-2 knockdown in C. elegans with regards to lifespan and health span. Now, with regard to gene interventions for lifespan extension, model organisms have been a fruitful ground for testing these. Ever since Cynthia Kenyon's group's seminal paper in the early 90s, many such genetic interventions have been described, both for positive regulators as well as negative regulators of lifespan and health span. These are genes which in general are highly conserved across taxa. They share expression patterns. They are in general involved in similar processes and consistently impact lifespan throughout various species. And of course, in keeping with this seminal landmark in the field, many of them have either are part of or in crosstalk with, of course, the insulin and insulin-like growth factor signaling pathway, or IIS, which in the nematode C. elegans is also known as DAF2, the DAF2 signaling pathway. So, these are, of course, all of us know a little bit about it. These are, of course, key regulators of metabolism, of responses to food availability, of endocrine regulation, and their knockdown in C. elegans has produced significant and substantial improvement of lifespan. Now, C. elegans is very amenable for aging research, for longevity studies, as in normal conditions, its lifespan at 20 degrees and regular feeding conditions, its lifespan is something around 18 to 20 days. So this means that in a lab setting, it's highly possible to do a full follow-up from larval stages, from synchronized larval stages up until death, and observe, of course, the behavior, the changes that occur with age, and of course assess them quantitatively to make value judgments on what happens. So because of this, of historical and biological reasons, the DAF2 pathway, the IIS pathway, is the logical entryway into prolongevity studies of any gene interactions. Now, as I've mentioned earlier, there's been a lot of pro-longevity and anti-longevity, longevity-associated genes, longevity-modulating genes, and interventions based on these, which have been described for model organisms and notably in C. elegans. However, a less studied topic is that of interactions between genes, of possible synergisms between these genes with regards to inducing a pro-longevity effect. And this is where the gerontomics project aimed to make a mark. Now, as our colleagues Vlad and Gabi previously discussed, one of the key goals has been to generate a pipeline, a bioinformatic set of tools for successfully predicting prolongevity gene interactions based on the breadth of data that we have accumulated through these mostly single gene interventions that have been conducted in the past. Now, of course, the following genes have been selected based on criteria. One of them, which I've touched upon previously, is a potential crosstalk with DAF2 silencing. Of course, they're part of individual clusters in the multi-interactome embedding, decreased number of shared KEGG pathways, and so forth. So there's been a number of criteria that have factored into the selection. And of course, the shortlisted genes were manually assessed by our team, of course, bioinformatically and biologically. And in the end, a set of 3 genes, odr-3, ife-2, and CKU70 were selected. Now, these genes, of course, they have well-known mammalian orthologs. For odr-3, it's the G protein alpha subunit. In C. elegans, it's involved in olfactory sensing. It's essentially found, its expression is found only in 5 pairs of sensory neurons. Essentially, they're olfactory mutants. These are the knockdown of odr-3 yields olfactory deficient animals. ife-2 is the ortholog of eif-4e, so an elongation protein synthesis related protein that is key also to homeostasis and stress responses. And CKU70, an ortholog of KU70, which is involved in DNA repair and various stress responses. Now, all of them, as I've briefly touched upon earlier, potentiate life-extending effects of DAF2 mutants. They have been described to interact in such a way. Now, the aims of the present study have been, of course, to assess the effects of combined interventions on these genes for C. elegans lifespan and, of course, for healthspan, because healthy living is just as important, if not more so, than absolute age. Of course, investigating crosstalks with the IIS pathway by DAF16 knockdown, and in the process to validate the bioinformatic pipeline of prediction that has been created and put forth by my colleagues, as described previously. Now, for clarity's sake, I will point out that all of the mutations I'll be referring to are knockdowns. So these are essentially genes that have been silenced. We're talking about gene silencing. And there's a number of ways in which this can be achieved in C. elegans. The main intervention that we've opted for was feeding RNAi. We've used the Oringer library, which is the default and most widely used collection of interfering RNA for C. elegans that's designed for feeding the worms. Essentially, the worms are plated on bacteria which have a plasmid that produces a double-stranded RNA, which silences the expression of the gene of interest. So using odr-3 and mutants and wild-type worms, we studied the effect of the odr-3 ife-2 interaction. And we found that for the double mutants, the odr-3 ife-2 combination of silenced genes, the effect wasn't on lifespan, was an additive one, essentially meaning that mean and maximum survival are roughly a sum of individual lifespan extension yielded by each of the two individual genes. CKU70, however, did not yield a similar effect, but instead it decreased the extension of lifespan conferred by the odr-3 mutation. So as you can see here, the double mutant is on average less longer lived than odr-3, than the single mutant. CKU70 RNAi was also shown to have deleterious effects on the double mutants. So as you can see in the figure on the right, the odr-3 ife-2 double mutant, which has been fed with CKU70, has a shorter lifespan compared with odr-3 ife-2 double intervention. The only interaction in which CKU70 was shown to have a mildly positive effect was its interaction with ife-2 alone, where it yielded only modest, statistically significant, but biologically questionable effects on lifespan extension, both medium and maximum. So with this in mind, the question arose whether these effects are mediated or not by the IIS pathway, as previously discussed. And we repeated essentially the same intervention, only this time in a DAF16 mutant background. So the thing is, with the DAF16 odr-3 double mutants and the DAF16 ife-2 double mutants, the effect that we found was, of course, not of course, actually, was surprisingly opposite, went in opposite directions. That is to say, DAF16 odr-3 was shorter lived than DAF16 background. So, as you can see here, whereas DAF16 ife-2 was longer lived than the DAF16 background. And of course, CKU 70 didn't yield any substantial effects. It almost closely mirrored the lifespan of DAF16 and DAF16 CKU. So it appears that the odr-3 ife-2 interaction is independent of DAF16. And again, even in this DAF16 deficient background, CKU 70 intervention did not influence lifespan. And even in the setting of the quadruple mutant, DAF16, odr-3, CKU, and ife-2, labeled here in purple, it closely mirrored the DAF16 single mutant. So no added effect here. In order to validate, of course, this finding, and especially the opposite directions in which the lifespan effect occurred for odr-3 and ife-2, we also attempted to directly visualize DAF16 nuclear translocation using a fused construct, a DAF16 GFP construct that is visible on confocal microscopy. We could only find this in intestinal cells and in the odr-3 mutant. None of the others showed nuclear translocation of DAF16, suggesting, as previously discussed, that the odr-3 ife-2 double mutant with its additive effect on lifespan extension acts in a way that is non-DAF2, non-IIS dependent. So, of course, we did these interventions with RNAi, but they also had to be validated with regular knockdown mutants. The trend was maintained to the extent to which odr-3 and ife-2 still exhibit an additive effect on lifespan. However, the magnitude of the effect, the percentages themselves of impact on lifespan, on medium and maximum lifespan, were reduced compared with RNAi. This is a phenomenon that has been described previously for other genetic interventions. It could be, it's a matter of speculation. It's actually an open scientific question why this is the case. It could be either that RNAi has a more global effect on the entire organism, or conversely that the mutant strain impacts some groups of cells which RNAi may not go to and lead to a less favorable outcome on the whole. Another interesting finding here was when validating the experiment without odr treatment, is that it was found that lifespan was increased in a significant fashion for all strains compared with the odr treated experiment. In general, in literature, odr appears to increase lifespan rather than decrease it. We suspect that this is a hormetic effect that has to do with the concentration of odr used. So it's a low dose stimulation, high dose inhibition type phenomenon. So with lifespan out of the way, the question of measuring healthy life, that is to say health span, arose. And in order to perform this, we first assessed sarcopenia, so decline of motility with aging. Now, the way in which we did this was we came up with a semi-quantitative scale. We graded all worms according to their motility. We gave a score of A for worms that had unencumbered motility. We gave a score of B to worms which had which could respond to prodding with a platinum wire, but couldn't move more than a 0.5 centimeter radius. And we gave a score of C to the individuals which were in a frail state. That is to say, they were alive, they had a response, a mild response to prodding, but couldn't move in place. So we've performed this for every assay that was done until the end of it daily. And we found, of course, that some interventions, more than others, had an effect on health span as opposed to lifespan. odr-3, for instance, which as a single mutant has the strongest effect on lifespan, did not reduce, at least percentage-wise, the fraction of impaired and frail state animals. ife-2, on the other hand, which has a milder effect on lifespan extension, did do so. And the interaction between them, the odr-3 ife-2 double mutant, increased the fraction of healthy state living and yielded a decrease in the fraction of frail state worms. So with this observation, we decided to quantify it more thoroughly using Kaplan-Meier survival curves. However, instead of plotting alive versus dead, we plotted mobile versus impaired and or frail. And with regards to this, it became clear, as you can see in the figure in the bottom of the screen, that the odr-3 ife-2 interaction with regards to health span, to the maintenance of motility state, yielded an effect that was fully synergistic compared with both single interventions with regard to this maintenance of motility as assessed by body movement. Now, another way in which age-related decline through motility can be measured is through pharyngeal pumping assays. That is to say, the pharynx pump of C. elegans has sort of automatism, built-in automatism, which exhibits a gradual decline with age. And it was recorded, and the number of pumps per minute were assessed. This is actually less intuitive than it seems, because it would be safe to say, for instance, that, because calorie restriction, feeding restriction could lead to improved outcome, one may have expected that a decline in pharyngeal pumping might be associated with a greater outcome. However, this was not the case. And for the odr-3 ife-2 double mutation interaction, a positive increase was detected even from day 5. So whereas the wild type and the single mutants gradually started to exhibit decline, the decline was much slower in the odr-3 ife-2 double mutant. And at day 15, when pumping was at a very low level, odr-3 and ife-2 yielded a 187% increase compared with wild type, and corresponded again to a fully synergistic effect, analogous to the one that we described for body movements. Another parameter of health span that need be assessed is resistance to stress, to oxidative stress and to heat stress. So with regards to oxidative stress, we opted for 2 strategies, that is to say sodium azide and paraquat. Paraquat, of course, generates reactive oxygen species. Paraquat sodium azide generates hydrogen peroxide. And as you can see in the figures here, while the process was largely incremental, at least for paraquat exposure, this cannot be considered to be either an additive or a synergistic effect. It's simply an incremental one. And again, for sodium azide, so for resistance to hydrogen peroxide, ife-2 and odr-3 ife-2 were in a plateau, so there's no difference here. There is an increase compared with wild type, which is very susceptible. However, it's not comparable with the effects described for sarcopenia-based health span. With regards to heat stress, there were no, we couldn't say that their silencing affects the heat stress response. Another issue that needed to be tested was the correlation with reproductivity. Oftentimes, fecundity and longevity are a trade-off, and we needed to assess this by counting the brood size during the time of reproductive maturity of the animals. So it's safe to say that there is no such process at play here, and that both the single mutants as well as the double intervention closely mirrors the wild type. So nothing of the sort. So a few key takeaways. Obviously, odr-3 and ife-2 exert an additive effect on lifespan and a synergistic effect on healthspan, at least with concern to sarcopenia. Their simultaneous silencing increases lifespan, doesn't affect fecundity, increases muscle activity, and maintains activation of stress response mechanisms. odr-3 and ife-2 have opposing effects in DAF16 backgrounds. Their interaction is likely, highly likely, independent of DAF16, possibly converges downstream of DAF16. And lastly, CKU70 knockdown does not increase the lifespan of single or double mutants, is deleterious to health span, and may be an intervention that becomes deleterious with increased age, given its negative impacts on maximum lifespan as well. So with that being said, I'd like to acknowledge my co-authors, Vimbai Samukange, Gabriela Bunu, Dmitri Toren, Simona Ghenea, Robita Kutu. This paper is in press at Aging Albany. And I would also like to acknowledge the other members of the team who have worked on the tools, the databases, the predictions, the bioinformatic analysis that made this work possible. And big thank you to Robbie for gerontonics and having us on it, which I think led to great things for everyone. Thank you very much. Open to questions.
Thank you, Ioan. If there are any questions.
Yes, hello?
Yeah.
Hi.
Hi, Kostin.
Sorry for joining so late. I'm not sure. Okay, maybe I missed the nature of the two proteins, but is it possible to modulate their activity by small compounds?
It could be. So actually, thank you for the question, Yunus. Actually, one of the future plans for our work is, and Gabi is handling it, she's coordinating this, it's part of her thesis work as well, is to find pharmacological compounds that may either interact with or mimic such genetic interventions. So yeah, this is absolutely something we're
are there any known modulators, chemical modulators of the two proteins?
Well, I mean, I'd have to look into it per se. It's possible that there are, but at any rate, you know, they have basically some phenotypic readouts. It would be fairly easy to test if we had any serious candidates in this regard.
As far as I know, we do not have drugs that single targeted affect these genes, and more general drugs, it would be a bit harder to prove the synergy. So for our experimental design, this was not the most appropriate direction. But of course, there are interesting experiments to carry out in which we combine drugs to get synergies between them or between them and genetic interventions. Is this answering your question a bit, or?
Yeah. It's but and the second one, is it possible? Have you done rescue experiments?
Sorry?
Have you done rescue experiments where you rescue the phenotype by?
Well, I would say the DAF16 experiments, even though they're not rescue experiments per se, function in such a way. And you can see, for instance, that ife-2 did increase the lifespan of the short-lived DAF16 mutant. It was not necessarily in our direct interest to conduct rescue experiments this time, but we make use of the tool for the purpose of at least partially elucidating the mechanism at play.
I would say that rescue experiments are more useful when you deal with short-lived mutants. You want to show indeed that the detrimental effect can be removed. If you look for interventions that extend lifespan, you want something that extends lifespan. So then you.
No, I was. I mean, rescue just to show the specificity of the intervention. Just to reverse the phenotype. I mean, it has nothing to do with...
We did some tests to see that RNA intervention actually works, so that it is specific, that it reduces the target. But besides that, I would say that extending lifespan just by chance is something very improbable.
No, I know, but this is the classical. I mean, when we are doing loss of function analysis, by CRISPR or by shRNA or whatever method of down regulating a gene or knocking down or knocking out a gene there are some off-target effects. So that's why people are asking, at least in cells.
So as far as we know, the RNAi from this library is quite specific. It has a very good affinity. We did test that the genes are reduced. It's hard to estimate if. Can you hear me?
So it's an RNAi, it's not a knockout.
Yes. We did both. So
it's a combination.
Yes. So we did double RNAi, and then we also did double mutants. With the mutants, we saw a smaller effect, but still in the same direction.
And the data are shown here, but we also conducted real-time PCR for the targets of DAF16. We did phenotypic assessments to check,
sure.
For instance, the odr-3 readouts, the lack of sensitivity to isoamyl alcohol. We did the thermotolerance assay for ife-2. So we tested in multiple accepted ways by the C. elegans community to make sure that
sure. No, that was
and the targeting occurred. Now, with regards to the genes themselves, PCR was not amenable due to the design features of the Aringer library, which places primers upstream of the UTR regions. This is something actually the reviewers asked us as well. However, all other tools that were at our disposal to fully validate phenotypically and molecularly that the interventions had successfully and specifically occurred, we have performed them.
The only thing that I would add is that, so you mentioned off targets. There is no way to prove that no other genes, so genes are all connected and they influence each other. So it is possible that these two genes are affecting other genes, and the effect might be extended also because of those changes. But you cannot go downstream too much to really see what is the cause. It seems that the direct cause is the reduction in expression of these two genes. And
sure, that is another issue. I mean, it's not the indirect, it's just the off-target effects of RNAi, and that is
of course the transcriptomics of these mutants is probably different than that of wild type, so a lot of changes happen there.
I know. No, it's just what is the workflow generally agreed in the C. elegans community. So that was the question.
In general, if you create a short-lived model, you really have to do a lot of due diligence to show that those genes are related, the reduction in lifespan is related to an aging process and not to some pathologies or something that you mess up. But when we are talking about lifespan extension, this is not so much of a problem because it's not easy to extend lifespan by chance, basically.